Hi guys, here's Eric. Today I'm going to introduce you to the process of meiosis. Well, basically, meiosis is a big process which consists of several smaller processes. It includes interphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1, interphase 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. Today we are only focusing on mosquitoes because human cells consist of 46 chromosomes, which is hard to draw, and mosquitoes only have 6, so it's much easier to draw on a piece of paper. The meiosis process begins at interphase 1, which most cell does their normal cell functions and they grow. And this is a special cell which makes 4 gametes. In the end, it is called a spermatocyte, which is for male, and oocyte, which is if it's a female cell. During interphase 1, the cell, this is the nucleus, the cell duplicates its DNA, which is essential for the process of meiosis. After the interphase, we arrive at prophase 1, where chromatins condense into chromosomes. They become visible inside the nucleus, and the nucleus membrane disappears. This is the centrioles, which will pull these chromosomes apart, and it has spindle fibers. This is basically the cytoplasm. This is the centrosome, like the black spot in the middle. Also, during the prophase 1, crossing over happens. Basically means a homologous pair occurs and their shared genes. You can see here. Just visualize them as little marbles, and they exchange them through the process of crossing over. They are all unique because, as you can see here, there's red and yellow marble, you can say, and then green, orange, and red, orange, and green, yellow. Neither of them are the same. They are basically different from each other because you can see their genes are totally different. The next process is called metaphase 1. Basically, what happens is that the homologous pairs of chromosomes line up in the middle by the spindle fibers of the centrioles. Next, of course, the spindle fibers pull those homologous pair of chromosomes apart by the spindle fiber just pull them to the very end, to the each end of the cell. Then that's where telophase 1 occurs. Basically, 2 nuclei form from those pairs of chromosomes. There's nucleus membrane, and also cytoplasm pinches off at this stage. After that, a process called cytokinesis happens almost simultaneously, where 2 new cells are produced from that single mother cell. And also it's worth mentioning that before interphase 1, there are 6 chromosomes in the cell, and also 6 chromatids. Like, each slice of the chromosome is 1 chromatid. And after interface 1, where it's copied, like every chromatid is copied. After interface 1, there are 6 chromosomes and 12 chromatids. And after metaphase 1, meiosis 1, ME stands for meiosis, 3 chromosomes are present. You can see 3 chromosomes, and then 6 chromatids are in those 3 chromosomes. Like each slice is one single chromatid. After cytokinesis 1, interphase 2 and prophase 2 occur, where cell develop, which is what most of them are doing right now, and centrioles copy itself. Remember, there's only one centriole in that single cell. Back, let me find the page. Back here, see, there's only one centrioles, but in interphase 2 and prophase 2, it copies itself. So there are 2 centrioles, and nucleus membrane disappears because the chromosomes need to move within the cell. After that, metaphase 2 occurs, where the chromosomes line up in single file. Remember, there isn't a homologous pair present, so they have to line up by themselves. In the middle, they're lined up by those centrioles and their spindle fibers, essentially. Then, during anaphase 2, they are pulled away. Then, telophase 2 occurs, which is very similar to telophase 1, where nucleus membrane starts forming and cytoplasm starts pinching off. Finally, we arrive at cytokinesis 2, where 4 individual cells are produced and chromosomes also uncoil to form chromatins, which is uncoiled DNA, and they are all haploids. They are all produced from a single diploid, which is just their mother cell. We just went over all the small processes in meiosis, but there's also appendix I want to go over, so let's just start it. So a male haploid cell is called a sperm cell, and female is a female one is called an egg cell. And remember the chart from before. So after meiosis 2, there are 3 chromosomes in the nucleus, and there are 3 chromatids, 3 leaves, you can call that, 3 leaves in the nucleus, as you can see above. And I also want to go over the reason why every gamete is unique. This is because of independent assortment and crossing over. So crossing over, as we just talked before, is where they exchange marbles, where they exchange genes. And then independent assortment is basically different chromatids can be assorted into single nucleus. It doesn't necessarily need to be blue and purple. It can be red and purple, or yellow with blue. So that leads to the variety in gametes, and it eventually leads to the variety in the zygote, which is us. So in mosquitoes, there are 2 to the power of 3 equals to 8 gamete differences. However, in human beings, the number becomes 2 to the power of 23 because that's how many chromosomes there are in a single gamete. So, like, every gamete is different from its siblings. For example, it can have red, yellow, and then red, blue, green, and then blue, red, blue, and then blue, yellow, green. So as you can see, every gamete is different from each other, and also it's different from its parent cell. It's from its parent diploid because it has half the number of chromatids than it does, and it has half the number of chromosomes than it has the starting cell, the starting special cell. Hooray! We get to the end. This is the bibliography. I found the source of my investigation from Amoeba Sisters, which is a YouTube channel, and BC Science Connections 9, which is the textbook. Okay, that's it for our meiosis investigation project. Thanks for watching the video, and hopefully I will see you guys next time. Bye bye.